I want to thank everybody for attending this, from walking from the front of the building in Seaside, Boston to here, which seems to be like in Brockton. So my name is Ian Thornell. I'm one of the co-moderators. My other co-moderator is Juan Ioanowski, and we have nothing to disclose. We've understood that impaired CFTR channel function causes CF, and we know a lot about this at the single cell level, and we know that specific CFTR mutations cause specific defects in CFTR. And this is even recapitulated when these mutations are introduced in heterologous systems. The level that we're going to talk about today is how does this CFTR dysfunction cause certain hallmarks of CF airway disease? And so I'm sure we're all familiar that the ionocyte has an abundance of CFTR in it. So on average, it's got a lot of transcripts. If you look across the other cell types of the airway, you can notice that on average, these numbers are very, very small. But if you start looking at the single cell level, some cells express just as much CFTR, and some express even more than the mean of ionocytes. And so this brings up the question, does cell type matter? And I think some of the talks today will start suggesting that it does. So I'm just going to do a quick exercise here. So if I have an object on my desk and I take it apart and I do analysis of all of its parts and I find that it has a 1 megaohm resistor in it, I can figure out that it's probably an electronic. And I know a 1 megaohm resistor is in a flashlight, so I'm going to say it's a flashlight. But this 1 megaohm resistor could also be in a walkie-talkie. And so just knowing one piece of the puzzle cannot tell you what it does. You have to look at the cell and the other things that are in it, and that context will tell you what CFTR is doing. So like the resistor in one thing, it can transmit light, and this resistor in a walkie-talkie transmits sound. CFTR in some cells might secrete bicarbonate or chloride, and in others absorb chloride. And so this model came from our group at Iowa, and one thing that we found is that on the basolateral membrane of ionocytes, they specifically express these chloride Barton channels. And this imparts ionocytes with the ability to absorb chloride in response to the lumen-negative potential difference across airways. And this is in the opposite direction of secretory cells. And so all this science is not being done in a bubble. Several other papers have come out in parallel, and we're starting to learn a lot more about this model. And there's going to be some conflict and some resolution. And so Juan and I talked, and I'm using his study as an example. So he saw that ionocytes can absorb chloride and secrete bicarbonate. Importantly, he saw no sodium flow over ionocytes. He goes, Ian, why you got ENaC in an ionocyte? I said, well, I put it there because of single-cell RNA sequencing, what I just told you guys you can't do. And so, his study suggests that ENaC's not there, but for chloride absorption to work, it doesn't matter where ENaC is. It can be on a surrounding cell. And so as we produce more data, we need to go back and think about those data in context of our own studies. And we're hoping that, the presentations of all these talks today will stimulate scientific conversation and refinement of models. So I think there's one major standing question and two sub-questions that really help get at that. And I think the major question, why we're all here, is which cell type do we need to target for gene therapy, right? So you have CFTR in all these cells. What is it doing? If I hit a basal cell, what does that do? If I hit a secretory cell, what does that do? If I restore only ionocytes, what does that do? But I think really to answer these questions, you need to go to basic science. And so how do ions flow through CFTR in these different airway cell types? And I think we're just at the tip of the iceberg, kind of nodding our heads saying that, well, maybe CFTR just doesn't secrete an airway. Maybe it can do some other stuff. And then the second question is, how do we tie these phenotypes to the hallmarks of CF airway disease? So today, we're going to go through some talks. We'll begin by kind of identifying the cell identities and which cells contain CFTR. We'll talk about associations of CFTR cell types and airway physiology. And throughout these talks, we'll highlight and discuss some seemingly conflicting data in the field. And hopefully this will kind of help us put together a more complete model of what CFTR is doing in the airways. And so, with that, we'll have Zoe Wisniewski come up from UNC, and she'll start orienting us to the different cells of the airway that contain CFTR.
Good morning. My name is Zoe Wisniewski. I'm a senior at UNC Chapel Hill, and I'm here today to present for Dr. Kenichi Okuda. And we're going to discuss the regulatory pathways associated with the distinct identities of large and small airway epithelia. Ooh, I am both excited and very nervous to talk to you guys. But I have nothing to disclose. And to begin our discussion, it's important to identify our small airways and what that really means. So our small airways are anything below 2 millimeters, and they consist of our bronchioles and terminal bronchioles, while our large airways contain our trachea and bronchus. Both regions are affected by cystic fibrosis. For instance, in our large airways, submucosal glands are dysfunctional. In CF, they have decreased volume and pH, but increased percent solids in their secretions. On the other hand, our small airways are a site of muco-obstruction, as well as a recent study from the Iowa group shows decreased mucociliary clearance in CF pig small airways. We are interested in the small airways today, and previous research and data has identified a specific cell type within the distal airway epithelia. We refer to these cells as distal airway specific secretory cells, or DAS. They have certain gene markers such as SFTPB and SCGB3A2. As you see in the histology staining, there's only expression of these genes in the bronchioles, not in the bronchus. Additionally, it is important to note the morphology of the SFTPB positive cells. They have more club-shaped, which indicates their secretory nature. And this cell population also goes by other names, depending on the publication. It's either terminal airway-enriched secretory cells or respiratory airway secretory cells. But either way, their genetic markers remain the same. And this is an interesting population because research has shown that they are lost in distal bronchioles in COPD and IPF. And recent data from our group shows that they are also lost in asthma. But how they are affected in cystic fibrosis remains somewhat unknown. But we believe that loss of DAS contributes to the pathogenesis of muco-obstructive lung diseases. So while there is no published data regarding how they're affected in cystic fibrosis, we believe that DAS are lost in the CF distal airways. Spatial transcriptomics shows that the DAS markers are downregulated in the CF tissue, and MUC5AC and MUC5B are upregulated instead. This data was then validated using IHC for DAS markers. As you can see in the normal cells, there's SFTPB and SCGB3A2 positive signals, but there aren't any in the cystic fibrosis. So our project has two aims. The first is to characterize region and cell type specific regulatory elements in the human large and small airway epithelium. And the second is to identify transcription factors specifically associated with distal airway secretory cells. In order to do this, we first had to validate that DAS are present within our primary human in vitro large and small airway epithelial culture models. We were able to do this by immunofluorescence. As you can see, there is SFTBP present in our small airway epithelial cells, but there isn't any in our large airway, which makes sense. And then next, we performed a single nuclei attack sequence. This assay uses an enzyme transposase TN5 that identifies global chromatin accessibility on a single cell level. And we were for instance, this attack signal plot shows that the enhancer is available for cell type X, but it isn't for cell type Y. This data was then integrated with existing single cell RNA sequence data to further subcluster our cell types. For instance, in this main UMAP, you see the general cell cluster types, and as well as the distribution of large and small airway epithelial cells. As you can see, they're roughly 50-50, large and small airway. And then the general subtypes were further subclustered to identify our population of interest, the 11 secretory or DAS. Interestingly, it's important to note that we weren't able to have as distinctive clusters when we were just using the attack sequence data, but the integrated data did show a distinct cluster. And looking at this plot, you can see that the DAS airways, like DAS cells, are mainly small airway epithelial cells, and their marker genes are more expressed within this population when compared to the other subtypes, such as TMEM45A and SCGB3A2. Next, using our chromatin accessibility data, we were able to identify region-specific chromatin accessibility in our airway epithelia. This was through looking at active transcription factor motifs in either our small or large airways. And these active transcription factor motifs were seen across cell type for each large or small airway. So NKX, GATA, and FOX are active in small airways across the board rather than cell type specific. But we did look at cell type specific transcription factor motifs as well. This heat map shows the top 50 motifs. And as you can see, there is differential expression and enrichment based on the cell type. For instance, our ciliated cells, no matter the subtype of them, are enriched in RFX associated with ciliogenesis. Our rare cells that include ionocytes, tufted and neuroendocrine, are enriched in POU and ASCL1. And the deuterosomal and the rare have the FOX motifs. And the basal have June, TEAD, and FOS. And then our secretory cells have these associated statin, NKX. We are most interested in secretory cells since DAS are secretory. And we were able to then look at DAS-specific chromatin accessibility and transcription factors. So this heat map is the top 50 motifs in our DAS population. And the DAS are identified at the top. And as you can see, most of the genes that are enriched within this population are also common among the secretory cells as a whole, except for this one specific region where it seems more DAS-specific. And these transcription factors include our NKX family. We were specifically interested in NKX 2.1 because it is most expressed in our lungs when compared to other parts of our body, unlike the NKX, the rest of the NKX family. Additionally, it has a known function for specification of small versus large or proximal versus distal during lung development. So we hypothesize that it also plays a role in DAS maintenance in the small airway epithelium. We were able to show that it is co-localized with our DAS markers using IF. As you can see, NKX 2.1 is co-localized with SFTPP and SCGB3A2, our DAS markers. And to further investigate NKX 2.1's role in DAS presence in the small airway epithelia, we used CRISPR-Cas9 to knock out NKX 2.1 in the LAE and SAE samples. And as you can see in the Western blot, the efficiency was pretty good as we have decreased protein expression for NKX in the NKX 2.1 knockout cultures for LAE and SAEs. Histology does show a morphological change as well. This being our negative guide RNA control, and this is our knockout. We had two controls, the non-electroporation and negative guide. But we also saw differential gene expression, more specifically in the small airways, as the whole mount for both the large and small airways were stained for MUC5AC, SFTPP, and A-tubulin. The large airways didn't show a significant difference in gene expression, from the negative guide control to the knockout. But our small airways showed a significant loss of SFTPP, our DAS marker, and an increase of MUC5AC, a goblet cell marker. So we wanted to investigate how this loss of DAS affects mucus, and we found that loss of DAS leads to abnormal mucus hyperconcentration. This heat map shows global changes in genes across large airways and small airways in both the negative guide and the knockout. As you can see, our goblet cell genes increase significantly in our SAE knockouts, while our DAS genes are downregulated. And this was then, we confirmed the presence or increase in MUC5AC and MUC5B using a Western blot. And as you can see, the NKX 2.1 knockout sample has more MUC5AC than like the negative guide. And the large airways were relatively unaffected by the knockout. MUC5B also increased, but not as, it was like a slighter difference. And then to further understand like just how the mucus is affected and how the membrane is, we did an Using chamber study to look at the electrophysiological properties of the mucus and the membrane. And what we found is that ENaC function decreased and CFTR remained the same. The ENaC function decreasing indicates that we have more fluid retention, which should theoretically thin the mucus and we would have more dilute secretions. But the CFTR remained the same because these were on non-CF samples. But interestingly enough, we performed a percent solids assay or analysis, and we saw that the SAE, like NKX 2.1 knockout samples, had increased percent solids despite having decreased ENaC function. So this indicates or suggests that the increase in goblet cells kind of overcomes the more dilute mucus, or decrease in ENaC function. We theorized that if we did that in CF samples, there would be a higher percent solids even than the NKX 2.1 knockouts in the small airways. So, in conclusion, we were able to identify differences in regulation between large and small airways, and we identified a specific transcription factor, NKX 2.1, that we think regulates the identity of DAS, a cell type that is lost in muco-obstructive lung disease, which we think contributes to the pathogenesis of them. And we believe that by repairing the mucociliary clearance system, by restoring distal airway identity, may be a viable therapeutic strategy in the future. And I would like to thank everyone in my lab, as well as all our collaborators and generous sponsors, and if you have any questions.
I have a question. So when you knock out the DAS, then you have a change in MUC5AC, but not MUC5B. Do you know why? Because the explanation for that, you said that the goblet cells will be producing more mucin, and that compensates for the fluid change. But wouldn't you expect to have both 5AC and B?
Yeah, so I think MUC5B did increase slightly, but not that much. But by knocking out NKX 2.1, you see increased goblet cell metaplasia, I think. And I think the MUC5AC is more of a marker for those goblet cells, maybe, and that's why it has more significant of a change than MUC5B. But I could be really wrong. So.
Yes.
Thank you. Very nice presentation and very nice data. I just have a technical question. So what's the difference between your small airway and large airway cultures? Is this just the origin of the cells, or do you culture them differently somehow?
The origin of the cells, yeah, based on where they were taken, because they're primary human cells and then expanded.
Okay,
but they were treated in the same way.
So the differentiation media is practically the same?
Yeah.
Mm-hmm. Okay, thank you.
Join me. Thank you, Zoe. Great question. Our next speaker is Feng Yuan from the University of Iowa. He will discuss the molecular regulatory pathway associated with the distinctive identities for large and small airway epithelium in humans.
My name is Feng Yuan. So this is my presenter disclosure slide. There are no relationships to disclose related to this presentation. My topic is characterization of ionocyte subtypes in the cystic fibrosis ferret, and I'm coming from Dr. John Engelhardt lab. So we're working on by using the cystic fibrosis ferret to dissect into the CF biology and pathology. So my talk will consist of 3 parts. So the part 1 will cover the ionocyte ion channel expression patterns. And part 2, I will talk about the ionocyte distribution along airways. And part 3, we will look into the pulmonary ionocyte subtypes in both normal and cystic fibrosis disease state. So part one, ionocyte ion channel expression patterns. The CFTR has been found highly expressed at transcription level in ionocytes as compared to other cell types. So here we examine the CFTR, the chloride transporter, the protein expression. We found out that the CFTR protein expression, they are specifically located at ATP61G3 positive ionocytes in a CFTR apical cap pattern. So this is from the wild type ferret trachea staining. The next we move into the double transgenic ferret Rosa TG FOXL1 Korea T2. So FOXL1 is a key transcription factor for ionocyte specification. So by using this double transgenic ferret, we're able to track ionocytes into the EGFP upon tamoxifen induction. So what we found here is the CFTR apical cap expressed in the EGFP positive ionocytes. So I think both of the ferrets species show that CFTR expression pattern, which is in the apical cap. Then next we examine more ionocytes. So we found some of the ionocytes have the double apical caps, as I show in the up panel, and some of them just have the single CFTR apical cap, while the function of the double cap ionocytes are still under investigation. Then to further confirm this CFTR apical cap specificity, so we generated a FOXL1 query ERT2 CFTR fluxed ferret. So by using this double transgenic ferret, we're able to delete CFTR gene specifically in ionocytes. So look at this slide. We found out that in a control group, without tamoxifen treatment, so we can find a CFTR apical cap in the BSND positive ionocyte in the epithelium. Then move to the tamoxifen treated CFTR conditional knockout ferrets, we found out the apical cap in a conditional knockout group is eliminated. So this further confirmed the CFTR apical cap specificity in pulmonary ionocytes. Then next we look into the intrapulmonary airway, some mucosal glands. We did CFTR staining. We found out the CFTR is highly expressed in those ATP61G3 positive ionocytes at some mucosal glands. So we can clearly see the CFTR apical cap pattern in ionocytes in some mucosal glands. So next, to further to study the CFTR expression in great detail, so we generated a CFTR EGFP transgenic ferret. So by which we inserted a LOXP splicing acceptor EGFP on the subside into the CFTR gene intron 1. So this ferret have two purposes. One is we are able to use EGFP to track the CFTR expression, and the other purpose is we are able to access the LOXP site by query, by which we are able to reactivate the CFTR gene in a CF background. So here's the diagram to show the ferret. So in the CFTR EGFP ferret, so which is a cystic fibrosis ferret. For example, if we generated a FOXL1 query ERT2 CFTR EGFP ferret, we're able to reactivate CFTR gene in an ionocyte-specific fashion. Then next, we check the CFTR EGFP signal in the epithelium. So we found out that the CFTR promoter-driven EGFP signal is very intensive, as we show in the image. And those CFTR promoter-driven EGFP showed a really good colocalization with the BSND positive ionocytes. And all those results really tell us how the ionocytes are highly expressing CFTR. Then next, we are looking at other ion channels, for example, the sodium-potassium pump, NKA. So as we know, NKA is a key player in source secretion in fish gill ionocytes. So then we checked the NKA expression in the wild-type trachea. So we found the NKA showed really good colocalization with the ionocyte marker ATP61G3, as we can see the ionocyte images from here. And we also found they showed a NKA like apical rings in the ionocytes. Then next, we just compared the ionocyte structure to fish. We found out both ferret and fish, they have this ionocyte apical ring structure and tubular extension. So we think the ionocyte structure are evolutionarily conserved. Then the other chloride channel is sodium-potassium chloride co-transporter. So we call it NKCC, so, which is required for chloride secretion. So here we look at the NKCC expression at protein level by using the immunofluorescence staining. We found out that the good colocalization in NKCC1 and ATP61G3 in pulmonary ionocytes in wild-type ferrets. So then we got into a conclusion that pulmonary ionocytes are highly enriched in ion channels, proton pump, ATP61G3, and chloride transporters, cystic fibrosis gene CFTR, and other ion transporters and chloride channels such as NK and NKCC1. So and then let's move to the part two. So part two, I will look into the ionocyte distribution along the airways. So the first place we look at is the most proximal part, which is the nasal epithelium. So we look into the nasal cavities. We use BSND as an ionocyte marker. We compare the nasal respiratory epithelium and the nasal main olfactory epithelium. So we found the ionocyte morphology is very different in the olfactory epithelium as compared to the respiratory epithelium. The ionocytes in olfactory epithelium showed a really long, like tubular extensions. And then we check the CFTR expression pattern. So this is a confocal image. We can clearly see the CFTR apical cap pattern in the BSND positive ionocytes in nasal epithelium. So then we want to compare the different places on the ionocyte density. So first we compare the nasal respiratory epithelium and the tracheal epithelium. So one is we found the ionocytes are really abundant in both nasal respiratory epithelium and the tracheal epithelium, so it's not as rare as we thought. Then next we got into the quantification. We found out that the BSND positive ionocyte per millimeter square in nasal epithelium showed a much higher ionocyte density as compared to tracheal epithelium. Then we moved on into proximal trachea and the primary bronchus. So here we use another ionocyte marker, ATP61G3. So what we found here is they show pretty similar ionocyte density when you compare proximal trachea to the primary bronchus. Then next we divided the tracheal surface into three different places. One is the surface, airway surface over tracheinous muscle, and airway surface over some mucosal glands, and airway surface over cartilage. So we found the ionocyte numbers are very different. So the total ionocyte count is the highest in the airway surface over some mucosal glands as compared to the airway surface over cartilage and the tracheinous muscle. And the ionocyte numbers per area is much higher in the airway surface, which is over some mucosal glands, as compared to the other places. So then next we move into the intrapulmonary airway, go to the secondary bronchus. So we check the ionocyte marker ATP61G3. So what we found here is those ionocytes are in a cluster pattern, and it really located at some mucosal glands in a secondary bronchus. So here we got into a conclusion that ionocytes appear in a proximal to distal gradient pattern along airways in ferrets. So the third part, I'm going to talk about some of the ionocyte subtypes we found at normal and cystic fibrosis disease state. So our lab generated those CF ferrets harboring the G551 mutation, which we are able to manage the cystic fibrosis disease state by using a CF modulator. So here we use two ionocyte markers here. One is ATP61G3 and the other one is BSND. So in a wild type, by using the ATP61G3 staining, so if you compare the wild type to the CF, we can see the ATP61G3 staining goes away in a cystic fibrosis ferret, but by using the BSND staining, we can see the BSND positive ionocytes remains in the cystic fibrosis ferret. So then get into a quantification of ionocyte numbers per area. So we can clearly see the ATP61G3 ionocyte numbers decrease in the CF as compared to the wild type, and there's a low difference on the BSND positive ionocytes, which promote us to think about they should be two kind of ionocytes in proximal trachea. One is the ATP61G3 positive, and the other one is the ATP61G3 negative. Then we use the NKCC1 as an ionocyte marker to do a co-staining of NKCC1 and ATP61G3. We compare the wild type and the cystic fibrosis ferret. What we found here is the good co-localization of the NKCC and the ATP61G3 in the wild type trachea. But moving to the CF, we found different populations. One is the NKCC1 positive and ATP61G3 negative ionocytes, and the other one is the NKCC1 positive and ATP61G3 positive ionocytes, as we showed in the image in the CF trachea. Then move into the much higher magnification, so we can clearly see the NKCC1 positive but ATP61G3 negative ionocytes in the upper panel. And then go to the bottom panel, we can find the two kind of ionocytes. One is the NKCC1 positive and the ATP61G3 negative, and the other one is the NKCC1 positive and the ATP61G3 positive ionocytes in the airway epithelium. Just go down into the intrapulmonary airway. So we want to figure out what happens in the ionocytes. So here we use ATP61G3 as an ionocyte marker to do the immunofluorescence staining. So then compare the wild-type ferrets to the cystic fibrosis ferret. What we found here is the ionocytes in a cluster pattern in a wild-type ferret intrapulmonary airway, so as we show here. And then moving to the cystic fibrosis ferret, so we can see the very huge expansion of the ionocyte numbers in a single cluster in the CF ferrets, and there are more ionocyte clusters in the CF ferret. Then getting into a quantification, we found out that in a cystic fibrosis ferret intrapulmonary airway, so those ionocyte numbers per cluster is significantly increased when you compare to the wild-type ferrets. And we also observed the ionocyte clusters significantly increased in a cystic fibrosis ferrets as compared to the wild-type ferrets. Then, as I told you, we have the ferrets. We are able to manage the cystic fibrosis phenotype. So here we just compare two kind of ferrets. One is the CFTR-G551D when it's self-modulated WAX770 off, which is a CF phenotype, and then compared to with modulated on. So we can clearly see those ionocyte clusters kind of reduced when those CF ferrets are on WAX770, which tells us the ionocyte clusters and the numbers really dependent on CFTR function. Then next we move into the BSND positive ionocytes. So we look at the cystic fibrosis airway. So here we check the BSND ionocytes. So we can see the BSND positive ionocytes also in a cluster pattern. And here we use the Catin-5 to enable the SMG collecting ducts. And so we can clearly see the BSND positive ionocyte expansion in the collecting ducts and in the some mucosal glands tubules. Then let's get into the summary slide. So my summary is: one, pulmonary ionocytes are enriched in ion channels, ATP61G3, and the cystic fibrosis gene, CFTR, and other chloride transporters and sodium transporters like NKA and NKCC1. And the other is pulmonary ionocyte really showed a pattern that is proximal to distal airway gradient. And three is ionocyte ion channel expression that defines the subtypes is very highly sensitive to the CFTR function. And my future direction is we're going to do some ionocyte lineage tracing in the context of the CF disease. I think by using ionocyte lineage tracing in the CF context, we're able to figure out more like ionocyte heterogeneity. Then go to the acknowledgement. So really be grateful to work in Dr. John Ungerhall lab, and thanks to the whole team, and also thanks my collaborator from Harvard Chan School of Public Health, Adam Haber's group, and the Harvard Medical School Annex. So I'm currently funded by the Cystic Fibrosis Research Institute. Then that's all my presentation. I will be happy to take any questions.
Hey, Fung. Great talk. Viral Shah, MGH. Beautiful, beautiful images despite the bad lighting in this room, you know? I had a quick question about the subtypes. How much of that is like immature or progenitor ionocytes rather than different types of the ionocyte, mature ionocyte itself? Or what do you suspect?
Are you talking about the progenitors?
Yeah, because you have the ions that you showed have different types of ion channel expression.
Yep.
Is that just a continuum? Are there some of those like immature ionocytes that are going to get that? I guess your lineage trace will get at that.
Yeah,
but
yeah.
any thoughts or insights on that?
Yeah, I think that's a really good question. The question is about like the ionocyte progenitors. So right now we think they're just one progenitor, which we call the real cell progenitor, can specify into ionocytes, but we're not sure how it changes under disease state, such as CF. But we're going to do some studies on that. Just want to profile those real cell progenitors in wild type and CF disease contexts to figure out. Yeah, but I think those are mature ionocytes because those are adult ferrets.
Okay,
Yeah.
cool. Thank you.
Yep.
I enjoyed your talk too.
Mm-hmm.
I am really interested in the subtypes of ionocytes that you were discussing.
Mm-hmm.
So do you think it's possible that there's CFTR-expressing ionocytes that have different complements of basolateral transporters? So do you see CFTR-positive ionocytes that lack NKCC1? So I'm just thinking, this relates to the maybe differential function of ionocytes depending on the complement of other transporters that are there?
Yeah, I think the question is about the CFTR expression and other like key chloride transporters such as NKCC and NK. So based on the staining technique, we don't have an approach to do that because some of the worked antibodies, they are in the same species. We're not able to get into that. But so in the future, so since we have a method to enrich the ionocytes, we're going to do more like single cell sequencing to sequencing ionocytes in more numbers and more depth. We're able to figure out the CFTR expression and NKCC or NKA at least at a transcription level. Hi, Wim.
Hi, Fang. Nice talk. I have a quick question about the nasal ionocytes. Have you seen any difference in the ion channel composition between nasal ionocytes versus proximal tracheal, distal tracheal?
Yeah,
That's my first question.
The question is about comparison between the proximal part, nasal cavity, and trachea. So some of the staining technique we did, I think they are pretty similar. I think both of the ionocytes from nasal epithelium and the trachea, they express NSBSND, HPCC1, G3, and CFTR. So I think one of the images I showed is there's a huge difference in nasal epithelium and olfactory epithelium. So the olfactory epithelium has really long like tubular extensions, and those ionocyte tubular extensions trying to communicate with some sensory neurons.
Oh, yeah.
They could be different, but we're going to do more work into that.
What about NKA and NKCC?
Yeah, I haven't got into NKCC, but I will look at it.
The other question is, you have seen them tapering from proximal all the way to distal. So you see more in proximal tracheas, more ionocytes, but less in the distal tracheas.
Yep.
Also, you see them more associated with the, or on top of submucosal glands. Do you think it's about less submucosal glands downstream that leads to less ionocytes distally? Or?
Yeah, I think the ionocyte pattern when it go from the proximal to the distal, when it gets into a distal airway, like intrapulmonary airway, there are more ionocytes in the submucosal glands than compared to the surface.
Thanks.
Yeah, thank you, Wim.
Hi, nice data. Do you know if inflammatory condition can modify subtype composition, ionocyte subtype composition?
Oh yeah, so this is a question about how we modify the ionocyte subtypes. So we previously published a paper on the subtypes. We think the ionocyte subtypes are really sensitive to the osmotic stress. So when you change the media osmolarity, it really alters the ionocyte subtypes. But we still need more evidence.
FOXI1 is also expressed in the kidney as well as the ear. Has the ferrets given any insight into why these organs that also have FOXI1 seem to be privileged in CF, right? So we don't tend to think of those organs as affected by CF lung disease, yet they express FOXI1.
Oh, I think Ian is talking about the FOXI1 like extensively expressed in the kidney. So in some of the FOXI1 knockout virus we have, they really show the kidney phenotype. So we can see the enlarged kidney and with a lot of fluid retention in the kidney. For regarding to the CF, I don't think there's a kidney disease in the CF. So we checked some of the CFTR function in the kidney. We check the CFTR function and we also check the CFTR expression. I think the CFTR expression is not like located into specific cell types. It's more like universally expressing all the kidney cell types.
Thanks. So maybe its regulation is different.
Yeah, the regulation
is different.
Yeah, mechanism and the function will be very different. Yeah.
I have a question. So you show, if I understood correctly, that in CF ferrets, there are ionocytes that express different types of transporters. But what does it matter in CF? They're not going to transport anything because they don't have CFTR. So are you suggesting that in CF, ionocytes have functions that are independent of CFTR?
Well, that's a good question. So question is about in the CF ionocytes, they still have other ion channels. Well, I think so. I wasn't sure about some of the chloride transporters, but for the like proton transporters, they may have a function there, I think.
So they turn into an acid production monster.
Yeah, acid production. I think it's the place you are working on, right?
Thank you very much.
Yep.
They're not monsters.
Thank you very much.
Okay.
I have one job, and it's to introduce people, and I introduced the wrong talk just now. I apologize, Feng. Come in. So now we're going to have the next speaker, Tarini Gunawardena. She is from the Hospital for Sick Children in Toronto, so Canada. And she's going to be talking about the progress towards studying CFTR channel regulation in different cell types in primary nasal epithelial cell cultures. So welcome, Tarini.
Thank you, Juan. Good morning, everyone. So I'm Tarini, and I work with the Hospital for Sick Children, and I'm a project manager there. So today, in our talk, we are planning to go through some of the progresses towards studying a CFTR channel regulation in different cell types in the primary nasal epithelial cultures. There are no relationships to disclose related to this presentation. So as we all know, the recent single-cell RNA-seq data has revolutionized our knowledge on the airway epithelial cell types that express CFTR. However, our understanding on the cell type-specific regulation of CFTR channel activity is still unclear. So the nasal epithelial cultures that I've been using in this presentation are generated as a part of the Program for Individualized Cystic Fibrosis Therapy program, which is done in collaboration with the Sick Kids Hospital and CF Canada, where we recruit patients. We have got about 144 patients recruited so far, during which we study their clinical history, and then we monitor their clinical parameters too. And in addition, we collect blood samples from these patients, and then we carry out whole genome sequencing. In addition, for a subset of these patients, we've generated iPSC lines. And we also collect the epithelial samples for these patients and then conduct RNA-seq studies. And we also generate the primary nasal epithelial cultures, which I'll be using throughout the presentation. And this biobank is also available for researchers. So if at all y'all are interested, feel free to contact the CFIT program. So the generation of the HNACs and the functional studies. So we have validated nasal culture models, which are differentiated into ALI liquid conditions in passage 3, using Using chamber studies. So this is a representative trace for the non-CF HNACs. I'm not sure whether you can see the cursor there. Yeah. Non-CF HNACs, where we see a good Foscolin 770 response. and then also a CFTR inhibitor response, showing that these nasal epithelial cultures are functional. And in addition, we also carry out Western blot studies. So this is a representative Western blot image from 5 of the non-CF donors that we have been studying, showing a good a presence of the fully mature band C, and then also the mild immature band C, band B, which is present in the Western studies. In addition, we have also conducted single-cell RNA-seq studies for a subset of 26 individuals, 10 of which are non-CF individuals, and 11 are actually homozygous for the delta 508 mutation, and then 5 of them which are homozygous for the tryptophan 1282 mutation. And in our cultures, we actually see that the presence of all the different cell types that we expect to see in the epithelial cells. So we do see the basal cells, and then we see the club cells, the ciliated cells, the rare cell population of ionocytes, and also the presence of brush cells. So we have also conducted immunofluorescence studies to confirm the presence of these different cell types using centrine and acetyltubulin for the staining of the ciliated cells, and also the presence of FOXI1 for the rare cell type ionocytes, and in addition SCGB1A1 for the presence of the club cells, and also the MUC5B staining for the presence of goblet cells. So moving forward, we also wanted to see what cells actually express the CFTR. So the first panel actually shows the expression of CFTR on a per cell basis. So as we all know, we see the highest CFTR expression in the ionocytes, and then we also see CFTR expression in the brush cells and the club cells, and it is important to appreciate that each dot here represents a specific donor, and there is a variation between the donors even though we are looking at different cell types. So the donor-specific variation is something that we have to take into consideration. And when we look into the sum of the expression of the CFTR using the collective CFTR expression, we actually see that the highest amount of CFTR is accounted for by the club cells, and moving forward with ciliated cells and then the suprabasal cells. However, the contribution from the ionocytes is quite low because of its rarity. This brings us to the aim of the study, which is to understand the cellular and molecular basis for regulated CFTR-mediated chloride conductance in the airway tissue, where our objective was to actually compare the different cell types, mainly the ionocytes, ciliated cells, and the club cells, specific regulation of CFTR channel function in non-CF epithelial cultures. And we hope that, as mentioned earlier during the session, that this will actually enable in development of effective therapies that target the relevant cell type in the CF airways. So looking into the markers that we have used during the study, we've used TMPR-SS11E, which was highly enriched in the ionocyte population, which was studied by the single-cell RNA-seq studies. We have also co-expressed, co-labeled ionocyte markers TMPR-SS11E, NGFR, and FOXI1 in fixed nasal epithelial cultures. In addition, we also did live staining using TMPR-SS11E, followed by the fixing of the cells and then co-staining with them with NGFR. And we have also labeled these cells, the ionocytes, with TMPR-SS11E in dissociated nasal cultures, which we'll be using for the patch clamp studies in the next slides. To identify the ciliated cells, so we have used the presence of a motile cilia. In addition, we have also transduced with GFP, and then we can observe the ciliated cells in them. However, the dissociated cells with motile cilia is what we have used for the Concord patch clamp studies. In order to identify the club cells, we've used PIGR staining, and we have co-labeled them with SCGB1A1, which was in fixed cultures. And then we have also conducted live cell staining for PIGR in the dissociated nasal epithelial cultures, which we'll be using for the patch clamp studies. So during our presentation, we have actually cultured nasal epithelial cells to passage 3 and differentiated them in an air-liquid interface for 28 days, after which we've carried out single-cell dissociation of these cells. And then these dissociated cells were coated in collagen-coated cover slips. So we collected the media in this, which was present above the adherent cells, which actually comprised of the non-adherent ciliated cells. And the adherent cells were stained with live staining, 40 MPR SS11E, and then patch clamp studies was carried out for these ionocytes. And then live cell staining was carried out for PIGR in order to identify the club cells, and patch clamp studies were carried out on them. The ciliated cell fraction that was collected was seeded in matrigel-coated cover slips, after which the patch clamp studies were carried out. The agonists that we have been using today is phoscolene and isoproterenol, and experiments carried out, oozing chamber experiments carried out by Gregory Lucas's group has shown that both agonists actually have the activation of the CFTR channel function. And then, in addition, there is the isoproterenol maximum activity can be brought up to the phoscolene activation. So for the patch clamp studies, we've used wholesale patch clamping, and then the cells were clamped at -40 millivolts, and a stepwise voltage clamping was carried out from -80 to 60 millivolts. And in all 3 cell types, the ciliated, club cells, and the ionocytes, we actually see that there is an isoproterenol activation, a phoscolene activation, and also that is inhibited well by the GlyH101. When we move further to study the differential stimulation of these different cell types that we have used, so we see that all the 3 cell types can be stimulated by isoproterenol and phoscolene. There was significant activation in phoscolene for the ionocytes. For the club cells, we see that there was a significant activation by isoproterenol, and then there was also a significant increase in the activation for the isoproterenol versus phoscolene. And interestingly, in ciliated cells, we see that there is a significant increase in the activation between the baseline and the isoproterenol. As we all know, so isoproterenol is a beta-androgenic receptor, which requires the nerve and the estrine complex to bring about its activity. And so we wanted to look into the estrine and the nerve complex presence, how it looked like in our single-cell RNA-seq data. So we did see that this complex is highly expressed in the ciliated cells and also in the club cells. In the ionocytes, we didn't really see the presence of estrine or the SLC9A3R1, which is also known as the NRF1 factor. So this, we have also looked into the presence of the estrine complex in the ionocytes and the ciliated cells. So we do not see the estrine present in the ionocytes even by immunofluorescence studies. However, in the ciliated cells, we do see that there is the presence of the estrine, the estrine complex in the ciliated cells. In the future, we actually want to conduct patch clamp studies on the cells from a patient with nonsense mutation to confirm that these currents were seen due to the CFTR channel activity. And we also want to understand the native regulation of CFTR in ionocytes and the other pulmonary cell types. With this, I would like to thank Dr. Christine Baer for the guidance and mentorship, and then the laboratory team, especially Kiara and Kai, who have been working on the immunofluorescence studies, and then Ling Jun, who has been working on the Western studies, and then Saumya and the Moraes lab for generating the nasal epithelial cultures, the Wong lab, including Tian, who has been very helpful conducting the patch clamp studies, and the John Parkinson's lab, Irina and Zoltan, for actually doing the single-cell RNA-seq analysis, the Gonska lab for carrying out the oozing chamber studies, and Dr. Felix Rajan and his team for the patient and the healthy individual recruitments, and our funding body is the CF Foundation, CFIT, the CFIT program, and Genome Canada, and all the patients and the healthy individuals who have been participating in the study. Thank you. And with this,
Thanks, that was a great talk. And so we're going to open up for questions, and we're doing great for time here. Thank you, guys. So we got plenty of time for questions.
Thank you. Very nice presentation.
Thank you.
Were you surprised to see similar functional responses in patch clamp studies between the different cell types? I mean, given the CFTR expression in ionocytes, you would expect maybe a higher response, right?
So so this was something that we are kind of looking at too. So we are not sure whether it's because of the donor variation or the different subclasses that are present within each cell type. So we do see a different difference in the activation of the channels, even though it's within the different cell populations too. So, yeah.
Tarini, Martin Mendel from the CF Foundation. Very nice talk. But I was also following up on the previous question. In particular, I was surprised about sort of the responses in ciliated cells because single-cell RNA-seq data and actually sort of functional data when we sorted out ciliated cells with Sotuba in our lab have kind of not usually suggested that there's much CFTR-mediated chloride transport, at least in an epithelium. Mm-hmm. So I'm wondering, in your patch clamp configuration, was that set up? Did you have a high chloride in the patch pipette so that you basically set up sort of an artificial gradient, or was it physiologic what you had in the patch?
So we used physiological conditions, and I think it was the bath buffer was around 300 osmolarity was around 306, and then for the intrapipette it was around 290. And the CFTR expression in the ciliated cells actually has been reported by Robert Terran's group too, so where they have conducted patch clamp studies and they see similar trends with the IV curves for the patch clamp studies, and they have been using the similar voltage clamping in their studies too. So that's something that has been reported previously too.
It remains a bit of a mystery because, for example, on Western blots we don't see any protein, whereas we can see quite a bit from secretory cells or so. But I guess we need to sort of work harder to figure that out.
So that's something that we want to try to, because we get a lot of ciliated cell population in our cell cultures. So the highest population is ciliated. So we want to kind of enrich the ciliated cell population later and then see it in other functional studies like the membrane potential assays and see how well we see a CFTR response there.
I mean, one little additional piece of data is actually like some years back when we worked with Jay Rajagopal, and so the dual SMART inhibition, we also used notch inhibition to basically make epithelial cultures which were 100% ciliated cells. And from those cultures, we had 0 CFTR conducted that we could stimulate with force column. So, I mean, just something to
think about.
Work on more, I guess.
Yeah. Thank you.
Hey, Tarini. So, great talk. I was wondering, so you're looking at current densities, which are normalized to picofarads. How do you account for the cells not having a uniform distribution of ion channels? So the capacitance is going to give you a good idea of the size of the cell, but what if that CFTR is really heavily concentrated in one area of the cell and that differs across the cell types?
Yeah, this is something that we have to work on. Yes, and patching is quite a task. Yeah.
Question, I think it's pretty stupid, but I did not understand why you tried isoproterenol.
Isoproterenol?
Yeah, why did you try isoproterenol? I didn't understand why was that different? Why you did for some NABMX and then isoproterenol? Why do you try isoproterenol?
So that's one of the physiological kind of activation methods, and then at the same time we want to try out different agonists like in our future studies too. So that is something in our to-do lists. Yeah.
And I have another question. So you use TMPRSS11E.
Yes.
So this is an antibody. Isn't the extracellular domain of this protein? Have you ever used it in tissue? In tissue. This is all cell culture.
In cell cultures,
Do you do it in tissue?
No, we haven't done it yet. But that is something that we have been thinking of, using lung slices to actually kind of replicate these studies and see what happens there. So that is still yet to be done.
Thank you.
Thank you.
All right, thanks. Joining, thanking, inspiring me.
Okay, and so next we have Javi Hajibabazadeh, and she's from the University of Iowa, and we'll talk about how the ionocyte contributes to ASL homeostasis.
Hello, everyone. My name is Javahir, and I'm a third-year PhD candidate in the lab of Dr. John Engelhardt, the University of Iowa Carver College of Medicine. The topic of my today's presentation is dissecting the contribution of pulmonary ionocytes to surface airway liquid homeostasis. So in the heterogeneous surface airway epithelium, it's very important to understand the precise physiological task of every cell type in order to dissect how these cells work to maintain the local homeostasis. And at different time points, researchers have been associating different diseases with certain cell types. So I'd like to focus on pulmonary ionocyte that has been associated with cystic fibrosis. Pulmonary ionocytes have been discovered in 2018 simultaneously by two research groups, and they identified that there's a rare cell type in human bronchus and in mouse trachea that co-expresses FOXI1, which is a master regulator of pulmonary ionocytes, and CFTR. In one of the experiments, they transcriptionally activated FOXI1, and then they measured the short circuit current. They found that the chloride secretion in group with FOXI1 transcriptional activation is higher than in control group. So the conclusion of these papers were that CFTR activity positively correlates with the ionocyte number, and even though ionocytes are rare cells representing only 1 to 2% of all epithelial cells in the airways, they are a major source of CFTR activity. However, the function of ionocytes has been subjected to a controversy by recently published papers. So one of these papers concluded that ionocytes mediate chloride in liquid absorption, and they did that by overexpressing FOXI1 and also by knocking out FOXI1 by CRISPR-Cas9. And then they measured airway surface liquid volume, the fluid absorption, and short circuit current. The conclusion was that in ionocyte-depleted FOXI1 knockout cultures, the absorption was less and the ASL volume was significantly higher than in controls. And the opposite is observed in FOXI1 overexpressing cultures. Before I go to the next slide, I'll briefly talk about BSND, or Barton, CLC, and K-type accessory subunit beta. So there are CLCKB and CLCKA chloride epithelial channels that are highly expressed in inner ear and in kidney. And both of these channels are very important for maintaining the local homeostasis, and they're both being activated by BSND. When ionocytes have been discovered, single-cell RNA sequencing data showed that ionocytes are enriched with BSND. That paper that I was talking about previously, authors of this paper, they also knocked out BSND with Cas9, and they overexpressed BSND and CLCKB. And they found that ionocyte-specific BSND and chloride channels contribute to liquid absorption. Another paper that was published by our lab suggested that ionocytes are implicated in both chloride secretion and absorption, and they did it by measuring the ASL height over a period of 24 hours. And in wild-type ferret airway cultures, we can see that there are absorptive and secretory phases that are followed by homeostasis. And these phases are lost in FOXI1 knockout and CFTR knockout cultures. So this paper suggested the implication of ionocytes in both secretion and absorption. And based on all this previously published papers, our hypothesis is that ionocytes are important for both anion and fluid secretion and absorption. In order to test our hypothesis, we deleted CFTR specifically in ionocytes by using triple transgenic ferrets expressing FOXI1 CRE-ERT2, ROSA-TG, and FLOX CFTR. We harvested the airways, and then we expanded total tracheal cells. And then we plated the stomatopositive total tracheal cells in organoid cultures and LI cultures. We differentiated them for 14 days, and then we started the induction with 4-hydroxytamoxifen in order to clip tomato and CFTR specifically in ionocytes. So as you can see here, these green cells are ionocytes with deleted CFTR. Then at day 21, we used organoid cultures for assessment of first colon-induced organoid swelling, and we used LI cultures for measuring the ISC in Using chamber. Our data from ISC measurements demonstrated that the cultures with ionocyte-specific deletion of CFTR under symmetrical chloride buffer have significantly reduced forskolin-induced CFTR secretory activity as opposed to control group. And then we decided to repeat the same experiment, but under low apical chloride buffer. And we can see that, again, in cultures with ionocyte-specific CFTR deletion, we have less chloride secretion as opposed to control groups. After that, we switched buffers, and we measured the current under low basolateral chloride. And here, again, there's a significant difference between these two groups, but forskolin induced not chloride secretion, but chloride absorption in both groups. And chloride absorption in response to forskolin was significantly reduced in cultures with ionocyte-specific CFTR deletion. These experiments conclude that CFTR of ionocytes is important for mediation of anion secretion and absorption. And these green arrows are just pointing that the readings of current are different in all three cases, and the reads are much higher under low apical chloride versus symmetric chloride and low basolateral chloride. Then our organoid swelling assay, as you can see, these are the wild-type cultures and cultures with CFTR deletion, specifically in ionocytes. On the left side, you can see the video, and on the right side are the images of the same video under at three different time points. So there's an obvious difference in wild-type cultures between 12 hours and time point 0, but there is an obvious difference in cultures with CFTR deletion. We quantified the organoid swelling, and we also had controls that have been induced with DMSO instead of amiloride and forskolin. And results of our quantification show that the wild-type organoids swell significantly more than the cultures with deleted CFTR. In our next experiment, we deleted BSND using H11Cas9 and LEF1 knock-in mouse model, and we didn't do anything with LEF1 in this experiment, and the reason why we used this mouse model is because of the Cas9 and GFP expression. We harvested airways, and then we expanded GFP-positive submucosal gland cells, or SMG cells. And then we co-transfected these cells with guide RNAs against BSND and GFP, sorted for GFP-negative cells, and then expanded this GFP-negative BSND knockout cells. We plated them on ALI cultures, differentiated them, and then measured the ISC in using chamber, and then we stained and imaged with confocal microscope. So the results from ISC measurement under symmetrical buffer demonstrated that the cultures with BSND knockout have significantly reduced chloride secretion versus wild-type SMG cultures. So we conclude that BSND in ionocytes is important for anion secretion. Here's the immunofluorescence staining validating the knockout of BSND. We stained for CFTR, which is in red, BSND in green, Hoist, which is blue, and FOXI1 in white. In wild-type cultures, we see triple positive cells, and we can see that there are double positive cells, which are CFTR and FOXI1 positive cells that lack BSND. The conclusions from our study is that ISC measurements and organoid swelling assay demonstrated that pulmonary ionocytes are important for mediating both airway fluid secretion and absorption. And surprisingly, we found that BSND is important for chloride secretion by ionocytes derived from mouse tracheal progenitors. With that, I would like to thank our funding sources and my PI and my lab for help and support. And I would like to thank you all for your time and consideration. And I'd be happy to take any questions. Thank you.
Thank you. We have plenty of time for questions, so feel free to walk on up. And
I enjoyed your talk, and I think it's we're starting to see the philosophy that the context of CFTR and these different cell types is really important. Do you think you would see the same effect on CFTR-mediated secretion by ionocytes if you knocked out the NKCC, as you did with the BSND? Is that something that's in the works?
That's a very interesting question. We haven't tried that yet. We're planning to. And I think that results might be the same as we see with BSND knockout.
While I'm here, I just wanted to respond to some of the questions about the ciliated cell patching. We were surprised to see that, and that's why we started to interrogate the molecular partners in the different cell types. Because if you're as old as I am, you know that there was a lot of discussion about the molecular, macromolecular complexes which enabled CFTR to work. So I think we need to think about the neighbors for CFTR in these different cell types. And it was only the ciliated cell where we saw appreciable levels of this macromolecular complex. So although we can't see the protein, that's true. The only signal we see in immunofluorescence is on the ionocytes. But CFTR is nestled in signaling proteins that may, even though the abundance is low, enable its activity.
Yeah, in a way, the proof is in the pudding, right? The channel function is the channel function. The antibodies have limitations too. Yeah.
I have a question for you, Java. So you're using, these are FOXI1 knockout ferrets, right? Yes, the question I have is the following. In the paper that you published earlier, the FOXI1 knockout was reported to have abnormalities in protein expressions in club cells and ciliated cells. And there were abnormalities in expression in those cells. So my question is it possible that part of the phenotype you observe is due to changes in other aspects of the epithelial? For example, what happens if you have abnormalities in a different cell type that you haven't investigated, cell, for example, ciliated cells, or if you have a change in paracellular pathways? If the paracellular pathway changes, some of the aspects of transport could be modified and would not necessarily be BSMD or ionocyte. It could be explained partially by other cell types. Is there any evidence that other cell types may also be working abnormally in this preparation?
You mean in FOXI1 knockout?
In both. When you modify, if you modify a transcription factor, it is possible that you affect other things during the process of development.
I think it is
my question
possible.
is, so do you have any evidence? Have you looked into other cell types?
Well, it's known that when ionocytes are knocked out, there is an increase in expression of secretory cells, because secretory cells also express CFTR, even though at low level, but they also increase the expression of CFTR. And we can see that there are more secretory cells in those cultures. And I'm sure that when we're knocking out pretty much any channel, there should be another compensatory mechanism that's going on in the culture.
So I have a question. The absorption assay data is really neat. It seems like ionocytes absorb and then they overshoot almost. They do their job too well, and then they make up for it and they secrete. Do you have any? Have you guys looked at why that switch happens? So what's going on at that 4-hour time point that they go from these cells that absorb to these cells that secrete?
No, so far we only have data about switching the chloride buffers. So we saw that ionocytes are absorbing under low basolateral chloride buffer. And we don't know how the switch happens yet.
Okay, well, thank you.
Thank you.
Oh, there is one. I'm sorry. No, come on up. Come up. Yeah, we got plenty of time.
It's like a follow-up about the knockout of FOXI1 in culture from the beginning. So at some point, we did experiments about knocking out FOXI1, but later on. So we used CRISPR-Cas9 to knock out FOXI1. And we didn't see much change in other cell types. But however, there's an ultimate experiment where you can make a suicide construct in cells that are expressing FOXI1. And we are thinking actually of doing something like expressing caspases and FOXI1 expressing cells. And this means we can turn it out, if we have FOXI1 cree and we can have stop locks upstream of caspase expressing construct, we can then activate this cell suicide program at a certain point. So we're planning to do that. We might be able to, after maturation of the culture, we can just add fluoroxamoxfen and kill only FOXI1 expressing cells. And this will answer, hopefully, ultimately answer your question. Thank you.
That would be great.
Okay, so I'm going to introduce our final speaker for this session, is Xiaojie Luan. He's from the University of Saskatchewan. and he's going to describe a novel relationship between MUC5AC positive cells and ionocytes.
Morning, everyone. Thank you for the introduction, Ian. And so I have no relation to disclose. So as you hear this morning, that pulmonary ionocytes show very high expression of CFTR, but it's a really rare cell type, only count about 1% of cell in the epithelia in the lung. However, the abnormal in ionocyte function have been proposed to contribute to the development of CF lung disease. So it's pretty amazing that 1% of cell can cause CF. So after the discovery of pulmonary ionocytes, the ionocyte function have been intensely investigated. So Dr. Okuda and Gensch summarize the current finding of ionocyte function here. So Feng and John Engelhardt group find that ionocytes can contribute to the secretion and absorption of liquid in the airway. And Ian's lab find that ionocytes contribute to the absorption of liquid in the airway. However, our recent data published on Blue Journal suggests that actually ionocytes are involved in bicarbonate secretion. And in that paper, we use pulmonary human bronchial epithelial cell culture. So we are wondering what happened with the function of ionocyte in a native tissue. So it has been suggested that the CF lung disease actually starts in the distal airway, where the 2 millimeter diameter airway that here. And to protect the airway, people believe the origin of the CF lung disease is because of the abnormal air surface liquid. And one important part of the airway surface liquid protecting mechanism is the mucin in the airway. So the mucin unpacking require appropriate pH and bicarbonate concentration. Dr. Quinton's paper show that the distal airway have a really unique anatomy. So it have pleat at the bottom of the distal airway and the fold at the top of the airway surface. And they also suggest that they show that the NACFC expression in the distal airway at the pleat and at the fold are different. So it suggests that maybe there's a different cell located at different location of the distal airway. So I'm wondering, our lab wondering where the ionocytes locate in the distal airway. So we hypothesize that the ionocytes secrete bicarbonate facilitate the unpacking of the mucin secreted by the mucin 5AC positive cell, which help protect the airway. However, this mechanism may fall in the CF airway. So our goal is to find where the ionocyte and the mucin 5AC positive cell locate in the distal airway in a native swine distal airway tissue. And then we want to know the function of the ionocyte in the native swine distal airway tissue. So first, we want to figure out where the ionocyte locate. So we dissect fresh distal airway tissue from swine, wild-type swine, and we do immunofluorescence study. And we mark mucin 5AC, and we use BSND and FOXI1 as marker for the pulmonary ionocyte, and followed in to mark the cell in the distal airway. So this is this is the dot stack we got. We move from the pleats of the distal airway to the fold of the distal airway. So as you can see, when the focus plane of the confocal microscope screen the distal airway, the ionocytes show up at the folds of the distal airway of native swine tissue. And then we further confirm this use slice of paraffin embedded swine distal airway tissue. As you can see, most of our ionocytes seem to locate at the fold of the distal airway of swine tissue. And we quantify that. We set the top of the fold as 100% and the bottom of the pleat as 0%. And you can see that the distribution of ionocyte from the pleat to the fold of the distal airway. As you can see, and we quantified 229 ionocytes from several slides we got from different peak and different region of the distal airway. And we find that almost 60% of the pulmonary ionocyte with BSND and FOXP1 labeling are located about 75%, so top 1/4 of the fold region. However, only 15%, about 15%, locate about 50% below, so in the pleat region. So, and the mucin 5AC positive cell are distribute along the way, and at the fold and the pleat and in the middle is distribution differently as ionocyte. The second part of this talk, we want to investigate the function of ionocyte in the native swine distal airway tissue. So how to find ionocyte in a native tissue? So we adopted the method that we publish in the Blue Journal using the pulmonary human bronchial amphibian cell culture. So we use a metal tracker to label pulmonary ionocyte. So pulmonary ionocyte is known as mitochondria-rich. So we incubate the native tissue with metal tracker. And then we find the brightest metal tracker labeled cell in the native tissue, distal airway tissue. And we measure ion transport, use our single cell level ion transport electrophysiology technique. And then after the finished measurement, we fix the tissue, mark the location of the cell we measure, and we use BSND as a marker for ionocyte. As you can see that the BSND marking is superimposed with the metal tracker image. However, you can realize that this method is not perfect. Some ionocytes didn't show up in the metal tracker imaging. So the technique we use to directly measure ion transport is called self-referencing ion selective microelectrode technique. And this graph demo how it works. So when an epithelial cell consistently transport ion from one side to another side of epithelial layer, in this case sodium, it will create an epithelial layer close to that cell. And when the cell consistently transport sodium from apical side to the basal lateral side, in this case, it will create a lower sodium layer close to the tissue compared to the background. So this ion selective microelectrode can pick up the gradient different close to the tissue and compared to the background. And we use Franck's laws of diffusion, can calculate back how fast the ion pass through that single cell. So we dissect our distal airway tissue from the swine, and we flatten it, and we use metal tracker label it, and then we use our technique to measure single cell level ion transport through pulmonary ionocyte in swine distal airway. So here's our results. We measured both proton movement and sodium movement through the ionocyte. And the y-axis show the ion flux, how fast it's moving the ion. And the positive number indicate that ion is actually moving from basolateral side to the apical side. And the negative number is from the lumen side to the basolateral side. And we stimulate CFTR with forskolin and MMX at time 150 seconds. So before any stimulation, we find that there's a acidification of happened to the lumen site when there's no stimulate happened for the pulmonary ionocyte. So it's a positive number. However, when we apply forskolin and MMX, it reverses the direction of the proton flux to calculate there. So it's moved from lumen side to the basolator side. It causes alkalinization of the airway lumen. And this can also caused by the secretion of bicarbonate. On the other hand, the sodium flux, there's nothing happened before any stimulation and forskolin IBMX stimulation through the pulmonary ionocyte. And then we wondering what happened in the CF tissue. So if CFTR is not working, so we incubate the native swine distal airway tissue with CFTR inhibitor 172. And we find that if we block CFTR channel, as expected, forskolin and IBMX didn't stimulate this CFTR-dependent pulmonary ionocyte function. And we quantify that, use area under the curve. We calculate how much the ion flux happened after forskolin and IBMX stimulation. So clearly, you can see that inhibitor 172 block this ionocyte function. And we are wondering whether the bicarbonate play that role to the pH regulation through ionocyte. So we block the carbonic hydrate function with azothodolomite. We incubate the tissue with azothodolomite. And the results show that azothodolomite kind of block the ionocyte function. So we think that this result indicate that the ionocyte seems to be secreted bicarbonate. Then we are wondering whether we can block that bicarbonate transport. So we use DIDS, block the anion channel exchanger 2. That we, in our blue journal, we think that is collaborate with CFTR to provide the ionocyte function. So our results show that incubate the native tissue with DIDS, it blocks the ionocyte function. So we think that the pulmonary ionocyte play a role in bicarbonate secretion in the distal airway. Then we do some functional study. We want to see if we block pulmonary ionocyte function in native tissue, what happened. So we use fluorescent recovery after photobleaching. So we collect the mucus secreted in the distal airway. Both control and DIDS incubate. And then we collect the mucus, we put FITC in the mucus, and then we use powerful laser to photobleach that FITC there. And we test the recovering process of the FITC in the photobleached region. So the recovery time indicates the viscosity of the mucus we collect. The longer the recovery time seems to indicate that the more viscous of the mucus there. So this is the intensity recovery curve, and we calculate the half-time of recovery. So our results show that if we block the ionocyte function with DIDS, there seems to be an increase of recovering time. So we think that the blocking the bicarbonate transport by ionocyte make the mucus more viscous. We also do another functional study that blocking ionocyte with DIDS, and we use mucociliary clearance assay to test how they affect the mucus clearance in distal airway. So here in the movie, we label the mucus bundle with acetylene blue, and we use titanium discs as an indicator as mucociliary clearance as well. So we calculate how the speed of those mucus move. And the results show that actually, if we block ionocyte function with DIDS, it reduces the speed of mucociliary clearance in the native swine distal airway. And this effect actually both mucus clearance and the flap can be recovered if we add bicarbonate on top of the distal airway. So in conclusion, pulmonary ionocytes form clusters at the fold of the small airway in swine. And our results indicate that pulmonary ionocytes play a role in bicarbonate secretion in distal airway. And the bicarbonate secretion from ionocytes may contribute to the unpacking of mucin from the mucin 5AC positive cells. And finally, I would like to thank our funding agent and then our leaders for our research group, Juan Anosky, Julian Tam, and Veronica Capinci, and all the lab members in our lab. Thank you. And any questions?
Thanks. We have plenty of time for questions, and we'll still get early for lunch. So it's a win-win. It's NACFC first.
So I love the methods that you described. It's so beautiful. At the beginning, you mentioned that you're using MitoTracker to identify the ionocytes. And then you also looked at labeling of the BSND. And there was good overlap, but it wasn't
perfect, yeah,
complete. And there may be some cells that are labeled with the MitoTracker that are not BSND, or the other way around, I can't remember. So what do you think those other cells are?
It's a really good question. So we are currently investigating what's happening there, and we, of course, use your lab's technique, maybe a solution for that. The label with TMPR11, SS11E, it's maybe a solution for that. And currently we don't know what's happening. And so we also play some catch-up game with Ingerhard group. There may be subtype, there may be not. We are not sure.
Yeah.
So the airway surface liquid is very thin, and the bicarbonate diffusion would be slow. If you calculate the diffusion, does that explain the distance between different ionocytes in an airway epithelia? Is an ionocyte, 1 millimeter away from another ionocyte sufficient to alkalinize the whole epithelia?
It's a good question. Yeah, I didn't do it. Yeah, so, yeah, it's a good question. Yeah.
So Jay, I have an additional question. So when you're looking at proton flux and you activate with Forceable and IBMX, the proton flux seemed to decay. Is that a breakdown? And I mean, this kind of dovetails to Joe's question. Is that a breakdown in the electrochemical gradient, or is that something else?
Also, the way that we calculate the proton flux is we calculate the total flux. So it can indicate the direct proton flux, and it also can indicate the bicarbonate. So we still need to figure out a way to separate them. It's better to separate them, but we are currently...
So they're inverse functions of each other. So my question is, why does it decay? Why doesn't it just go up to a level and stay there? Is it that the gradient's going away, or is it something else is happening?
If it's consistently moving the ion through ionocyte, it should stay there, but there's a chance that that Forceable IBMX have an acute effect, and then it's slowly decay the effect.
Yeah, but if you stimulate again, it comes back.
Yeah.
So it can be the gradient.
Yeah.
Hi, Scott Randell from the University of North Carolina, Chapel Hill. So phasic motion is an incredibly important physiological part of small airway biology, and others have described setups, for example, for precision cut lung slices to stretch them and relax them phasic. Have you thought about how that might impact with the changes in the ionocyte biology in the swine small airway?
Yeah, it's a good question, and I think Dr. Paul Quinton's paper suggests that it's like a washing machine, like expanding and closing and expanding of the distal airway. I believe that generally play a role, but we don't think that the fold and the pleat can switch. We think that it stays the same, and our immunohistology data show that it seems to be more located on the fold. So that's the reason why we conclude with.
Thank you.
We think.
I have a question about the ionocyte numbers. So in some of your images, you show that in a swine model in a distal airways, the ionocyte numbers seems like pretty high. So have you tried to compare the distal airway to the proximal airway? Is that like difference?
We have that data, but we haven't looked into it actually. It's if I remember it correctly, you are right that in the distal airway there seems to be more ionocyte there per millimeter square compared to the larger airway.
Okay.
But we haven't looked into it yet.
Yeah, it's like the opposite as we observed in a ferret.
Yeah.
So it's kind of species difference.
Yeah.
Is there? Yeah, there's one more question coming up, and we're still way under time. So don't you worry, guys. This is a great conversation.
Hi, I'm Carla Ribeiro from UNC Chapel Hill. Amazingly enough, over 20 years ago, we published a very interesting work showing that mitochondria polarize towards the apical domain of airway epithelia. Actually, mostly we were studying ciliated cells. And on those days, we thought about, well, why are the mitochondria polarized towards the apical domain? Is it because it's supposedly, giving a lot of energy to ciliated, movement, beating, things like that? But anyway, after this, I would like to ask a question. The overexpression of mitochondria in ionocytes. Question number one is, do you know if they're also polarized in ionocytes? And question number two is, what kind of function do you envision for this vast amount of mitochondria in ionocytes?
It's a good question. Like in our Blue Journal paper, we find same thing. It seems to locate at the close to the apical memory of the ionocyte. And I'm not an expert of mitochondria, but I think that the mitochondria, I'm not sure about whether the mitochondria is polarized or not. And the mitotracker that we use is mass-based. So the brighter the dimmer of the mitotracker didn't affect the polarization of the mitochondria. It's only affected by the mass of the mitochondria. And the reason we think that the mitochondria is really rich in ionocyte is we believe that the ionocyte is a very metabolic active cell. So, and I think Dr. John Ingerhardt's 92 paper already suggests that seems to show that the ionocyte is mitochondria rich, and so that's the reason why we use mitotracker.
Of course, there are several possibilities, but one possibility, because of the well-known function of mitochondria to sequester calcium when it's released from the ER, for example, or when it enters through the plasma membrane. So perhaps it's also participating, like we have shown in airway epithelia ciliated cells a long time ago. It participates in calcium sequestration, therefore calcium-dependent function, to restrict calcium-dependent function to the lateral membrane domain. And this would have implications for perhaps a lot of your work on transporters in the ionocytes.
Very good point. Thank you very much.
Okay, so if you have any other questions, we're going to finish our meeting. I want to thank everybody for participating and the speaker for a great talk and for the opportunity to organize this workshop.